Ladies and gentlemen, friends of ITU, thanks for coming. Also to you online, welcome to our first lecture. Today we have two very brainy ladies here who are going to present their research on explainable AI and computational neuroscience. And yeah, we're very glad that so many people could be here today. And I've just been informed that we have 10 professors from South America here, from the STEM network, educational technology. I'm very... Finland and just say it.
Serbia.
Peru, Indonesia, I heard. Yeah, so, well, we're very international today. Thanks for coming. I'm very honored to have you here, and I hope you very much enjoy today's talks. With this, I would like to introduce our first assistant professor, Nina Hubig, who has come to us with a background in computer science. She did her PhD in data mining in the Helmholtz Institute together with the TU München, and has from there on continued working a lot with biological medical data, except for a little stint at BMW, where she clearly worked on automotive, and then went to the US, to Clemson and Medical University of South Carolina, in order to do more of medical data science. And now she's here at ITU as our assistant professor for explainable AI. Let us welcome Nina.
So, hello everyone. I'm happy to be here, and today I'll give a joint talk with Jimei, and we decided on a topic that's called NeuroXAI, illuminating the black box of minds and machines. And the XAI is my part, the explainability of AI, and I want to start with a picture. So this picture, generated picture, Dolly. It shows this medical doctor, this lady, looking through this somehow floating thingy into a brain that's floating in a black box. And this black box has these interesting AI, circuits. And this is what I'm talking about. Today, I will talk about illuminating the mind of the machines that are for us, and this is where we basically connect the neuroscience and the explainability, that for us is both a black box, the real human brain and the artificial brain. If we look at it from the outside, we don't know what's going on inside. That's the black box part. Now, I ask you, what do you think of the mind of this robot? I had to bring this. I'm starting an AI professorship in Austria, and this is a very famous Austrian actor. And not just that, this is the gist of how the Western, but maybe the world, sees artificial intelligence in its dystopian view, right? This big, evil AI network, Skynet, that hijacks these robots, and then everything tries to kill humans. That's the meme of society since 1985. And there are other similar important virtual reality in this case, but behind the virtual real world reality, there is this machine system that tries to enslave humans. And a little bit more dystopian, but likely less known, this is the picture of the AI system of a Japanese animation called Psychopath. And you see a brain, because this is transhumanism. There's real human brains that connect each other. So each of these little things is a human brain. And if there's a decision, certain human brains come up like 5 or 10 of them, and you will see them making a decision together. But of course, they're not humans anymore, right? Together, they are this big AI system that are just depicted like that. And the name of it is pretty cool. It's in regards to this AI system working as a surveillance for humanity through something called a mental passport. So it generates a mental passport and it tells you, well, this is a criminal and this is not. So, this is how it works. Beyond dystopia, this is not what we want, right? We don't want a dystopia no matter how much society likes these kind of movies and likes it to think in this way. We want to have a controlled system. So what you have here is an autonomous driving vehicle. We can discuss how to control this is once it's level 5. But you basically see, well, this is seen as a human. This is seen as a different car. This is seen as like a Wi-Fi that connects how far away this car is, right? This is a robot comparison to the Terminator that is very controlled, that works in this controlled fashion, and it in this way is very usable as a tool. My research is in regard to effective and efficient collaboration between humans and machines, in particular in regard to high-stake decisions. So what's a high-stake decision? Come to it a bit later. These methods and I'm working is these interpretable models for decision-making, the reliable evaluation of it, its biases, and the effects of mental health. And this is not just one domain, of course, but the mental health domain would be more in healthcare, in law, in education and business would be other domains, as long as there is a high-stake decision. Explainability of AI, and this is where we are at. I'm coming from this machine learning direction, computer science direction. I add with the interaction the HCI and the explanation how human experts think a system works like. And the important part is the XAI is really this interface. It's about we as a human, we try to understand the system, right? This is not a system understanding the system. This is not something general. This is the human understanding the system. This is explainability of AI. When we talk in more factual, like models and particular black box models, we have to distinguish between two areas, like two dimensions. One is the accuracy dimension, and the other is the interpretability dimension, the level to which a human understands this particular machine learning system. And if you take a look into a decision tree, you understand a decision tree, right? You have a decision, a person is younger or older, you go this way, you go that way. This is fully understandable by everyone. You can understand when it will go left, when it will go right, and then when it comes to the leaves of this tree, where it will be. While if there is a neural network, these things that I depict as a brain, right, these artificial things, you don't know what's going on inside of it. So this is the typical black box problem that would need some post-hoc explanation. So another thing, what is this high-stake decision I was talking about? So I give here two examples, one in healthcare and one in criminal justice. In healthcare, we have the question for us that would be a typical high-stake decision, what treatment to recommend to the patient. And in criminal justice, I have this decision, should the defendant be released on bail or not, right? And both of these decisions have a significant impact on the human well-being, and that's what defines a high-stake decision. I gave you, I have a real-world example here for your real scenario from my research in medicine. This is about cardiology, about cardiac remodeling. So what is cardiac remodeling? So if you have something called hypertension, which is like basically high blood pressure, there is a scenario that's called cardiac remodeling. Your behavior of the heart, the functioning of the heart, the size of the heart changes significantly. Most often it grows bigger and bigger. And if that disease, that behavior of the heart, that change of the heart would persist, you would show clinical manifestations like arrhythmia, dysfunction, and in worst case, heart failure. So this is a typical, at the center, at the core of the body, there is this decision to take what to do now before you maybe even pass away. What do you do? What can you do? And as a collaborative tool, we have AI systems that can understand data to a degree that we simply can't, right? Thousands, millions of features, be it chemical features, biomarkers, mechanical biomarkers, functional biomarkers, thousands of them, age, the particular person, the precise person, what they had before, anything we can model. And this is something that's really, really excellent. And it helps us to do a very, very difficult decision, because there is a treatment for this cardiac remodeling, which is cardiac resynchronization therapy. And that's a relatively invasive therapy. But that's not the only problem. The problem is it only helps a certain amount of people. 2/3 of the patients will be treated and they actually have a heart that functions again better. And 1/3 will be treated and it won't help them. So if you can figure this out prior to doing the surgery, which is what we hope we can do with AI systems, then we are in a very different state of what treatment we can give someone, right? Or what we can do. So what we do now is we run an AI system, any form of classification. Here you see certain models, voting, classifier, stacking, classifier, up to adaptive boosting classifier. There is a training and a test data set. There are different types of feature sets. And we have 1,060 patients, which is for medicine, is a huge number of testing it on these patients, because the biomarkers, of course, and is very, very large data set for this. And let's take a look, well, let's take the perspective of the medical doctor. You would now have an AI system that tells you, well, I'm trained, I work, and now with this particular patient, he has a likelihood of 73% to benefit from the treatment. Does that help you? It might, right? It might, but it doesn't tell you why does that thing come to this conclusion, right? What's going on with that? So what we actually do with explainability of AI, despite this, what AI already can do, right? Figuring out with high accuracy what's going on, given all this information, we add the why. And the why on a global scale would be, well, there's a high responder and a low responder, and there is usually a high response on, for example, 1D stretch, which is one of the biomarkers. And this is the global sharp game theoretic method. And then there is this local for this particular patient. If he or she is a responder to the treatment, this is how these attributes look like. But if it's a non-responder, this is how the pattern will look like. And this information, once you are in this field, right, as I'm not going deeper into this, this will help you more, because it gives you a why. Why is this a good decision? Coming back to the societal level, there is a book, and you may have seen this already on in YouTube, because I was talking about this already a couple of years ago, and it's still relevant. It's relevant because this book explains how society, once we now roll out all these amazing algorithms, for example, in HR, this is the thing that actually came into my mind when I read in this book, how a school teacher never got a job again because this one trained algorithm was now rolled out in all the high schools she was applying, and they continuously dropped this one type of CV for whatever the reason, right? And this is something we want to mitigate, right? We want to mitigate systemic bias, and this is something we have to be very careful about. And this is very real, right, in comparison to the Terminator. Nevertheless, thinking a bit in arts, these are symbolisms that come up in AI continuously. And you see these two stretched out hands, and I guess most of you maybe know where this is coming from, right? This is a very old, here the lower picture is a very old image from Michelangelo from a church, the Sistine Chapel. And above we have the same picture, but with a robot arm. And this is how the whole picture looks like. If you haven't seen it yet, this is the divine arm. With all the help of the angels he can get, tries to reach the human who's like, yeah, maybe. And this is basically what we see here, right? The symbolism behind it is we reach for creating an AI system that has a certain form of human intelligence. And I mean, this is basically defined by a lot of, at this point, very wealthy companies that having even the name Singularity, which means a system that has this ability to code its own master code. And if this is super, like a human intelligent, maybe even more intelligent, and there's a lot of hype around it at this point in time. And no matter if it's hype or not, right, in the future, maybe in decades, we have to see that this is a direction people want to go. And we have to decide in research, how do we, what values do we want to bring in these systems that in the end, if it's an AI system that is such intelligent, it basically means we, can I say this? We need to, they want to go towards fully automated decision making, not just collaborating as a tool, but fully automation. And now we have to decide what values do we give such machines? What values do we have in our data, right? Which values would you choose? Here, the classic value for XAI, transparency and trust. Another value, contestability, empathy. What values do you choose if you think about it, right? In transparency and trust, very classic, there is a balance between the level of transparency that generates trust. Usually, if it's super transparent, it might not necessarily gain a lot of trust. For example, here for low transparency, you see a very sharp degradation the moment it becomes in regards to trust. But the moment it's very high transparency, you have the same issue, right? So there is a balance between low and high transparency. Too much transparency is not good either. You see this in regards to models. So this is, again, the decision tree. Here is the age, no, yes. And then you take a look at how much money the person has. And this is a high trust, high explainability method. The moment you add a high, the transparency stays the same. It becomes extremely complex. You cannot understand it anymore. And then it becomes problematic. Here with the neural network, that is the black box. You add some post hoc explanation. This is an image of a train. It should classify it as a train. Now you ask why? Well, because this is the important part where it's red, you know, these train things. This is what defines this train as a train, the heat map. A different thing XAI can do is something as called inbuilt model explanation. So we try to understand the domain, for example, neuroscience, through the lens of AI systems. You have here an fMRI scan, an image, and that image creates this 3D model with a certain postulation for brain regions. You have this time series data, you generate a correlation matrix of it, connectivity or adjacency matrix, and then the graph, the community structure. That's typical neuroscience research that Gia will later talk about it more. I want to focus here on the parcellation, the brain regions. This is an information we know already from neuroscience. Now, if we add that information in a neural network, for example, an extra layer that fires once a certain brain region is kept, we add information, extra information to a neural network it would usually not know, right? And with such help, the the researchers in this case. Another way to do would be to create a digital twin. In this case of the brain, could be anything, could be a city, could be what you want. And then you add knowledge. A digital twin is something like a simulation of that brain in this case. You add the explainability AI algorithms to it to understand how the inner functions work, which is one way to tackle this one, which is a black box and cannot be understood easily. And the thing I also did in regards to transparency is this is some form of transparency if you have a large language model and you don't want it to hallucinate because it's about claim checking. It's about science, it's about facts, right? And you add a database system that knows these facts, so it's working on ACID, and you have this LLM as an intermediator that runs simulations. From transparency, we have this concept of contestability. If I have expert systems and they run an algorithm system into the citizens, into the society, then there needs to be a feedback function that allows us to contest the system's decision. And this is basically what contestability is all about, the ability to challenge, review, and appeal a decision made by a system to solve problems like these weapons of math destruction decided. And here, the main reason besides user appeal, auditing, explainability, which is part of it, and is the human oversight. In explainability, when I think about how I would like to do it as an explainability method, the idea is to actually have the explanation done by two, for example, large language models that have a different persona, different worldview, and really try to fight each other. So we see different perspectives without needing to do it ourselves. In the end, what we want is this vibrant society. This city here and the sunrise shows this a little bit, right? And we do not need to be afraid of AI or XAI systems because in the end, this is like a mirror. The XAI system is a mirror that works like mirroring our what we are already. So in the end, if we run a system like this and try and understand it, we can choose who we want to be. And as such, we can shape our society the way we want. Thank you very much for your attention.
Thank you, Nina. Are there any questions already before we move to our second talk today about XAI, brains? Oh, sorry, didn't see you.
Thank you so much for the talk, Nina. I would like to hear a statement on you mentioned trust and transparency. So you said there is a correlation between trust and AI and transparency of the system. So if I think of my daughter going out on the weekend, that someday we will be there, I could trust her, or I could put a GPS tracker on her. When I make her behavior transparent using this GPS tracker, I think I'm not trusting her anymore. So maybe with people it's somehow inverse this relation, or what is the difference between trust in people and machines, or what do you think about it?
Well, actually, surprisingly, I saw work that says we trust machines more. And this was very surprising to me because when I have a person in front of me, I know how the person looks like. I might know some of the background of the person. I know the language. I know that they come from, right? I have a certain picture. But with the system, I usually don't have that, right? With the little daughter, it's a small child. Of course, you don't trust them to be safely around the, cars, right? Because they don't know. Maybe you don't need to have a GPS tracker for sure, but with a system talking to you, do you know about its persona, about its values, about anything? And despite that, people tend to trust them more. Why?
I want to throw in another term maybe here on trust, transparency, and justice, in a way that you made a reference to Cathy Neal, to the weapons of math destruction, and how algorithms sometimes create very unjust systems. And a lot of times the inference is made that transparency is the key to more just systems. Well, I'm not so sure about this, in a way that we have a lot of very transparent systems that are very unjust. They're openly unjust. So how do we value transparency, but also not overvalue transparency, and maybe connect to the issue of justice in algorithmic systems?
I would be interested in which system is overtly transparent and unjust, and then no one does something against it. Because, if it's transparent, usually transparency itself changes nothing, right? What changes something is when you can appeal it, right? When you speak up and say, this is not right. So if no one does this, that's the real issue, because maybe this transparent, unjust system, like maybe some form of dictatorship, right? There might be other mechanisms at play that don't allow a feedback function, right? So this is my thought about this.
I have no idea if I got part of this wrong, but you showed two figures with a relationship between trust and transparency, a low transparency, a high transparency, and the trust score dropped when you had a high transparency, right? So like the higher the transparency is, the lower the trust is in that figure on the right panel. Do you have any potential explanations why that's the case? Because it's kind of intuitively not making sense to me why higher transparency is not leading to lower trust score, or how is this trust quantified? That one, yeah, exactly.
So low transparency over like this, the self-grade, peer grade thing is a bit confusing, right? This is how they claim they trust, right? This is a user study, right? And basically there is a low transparency, and after a while, with certain tasks, right? They say they don't trust it as much. And it's part of it is because they believe it's wrong, right? They understand it enough to believe it's wrong. While here, the transparency is overwhelming, right? And there needs to be something in between. That's the core aspect here.
So can this be caused by the confusion due to high transparency, because there's so much information, it's kind of overloading people's mind, so they are more confused than they're supposed to be.
Exactly. Yeah.
With this, I think we're already on the right track. Jimé, we can hand over to you. Thank you, Nina. Yeah, let me introduce Jimé to you. She is our assistant professor for computational neuroscience. And she is the one professor who has lived in more different countries that I ever visited. So she's been really, really a mobile researcher. So this is very impressive. So she started her MS and then went to Berlin, to the Charité, in order to do her PhD, was in Canada as a postdoc, and then also shortly before she came here, she went to Tokyo, to Japan, as an assistant professor, and now she's at ITU and looking into the brain, and also trying to build neural networks which are brain-like.
Okay, so
Wow, it's really bright. Thanks for coming, and thanks a lot for the interest in our talks. And again, thanks for the introduction, Stephanie. So you have seen a lot of mini brains of mine across different places in the world. And it's a demonstration of the things I like, working in different countries, different systems, and use the brain power to understand the brain. So my talk is centering a topic of the synergy between brain and AI systems, specifically about how they can boost each other. So how can we use AI to understand the brain a bit better, and how we can use the brain findings and the knowledge we have about the brain to build better AI systems potentially. Yeah, that's pretty much my talk. But before we go on to the more complex topics, this is I was inspired by a professor in my master's because he's like, you're all in something related to neuroscience, but I really highly doubt that you know anything about the brain. So he started with this Neuroscience 101, brains and numbers, and he asked us questions, and no one could really answer them correctly. So I'm really asking you guys to rethink about this organ situated in the middle of your skull, and maybe just try to give some educated or random guess. The first question is a bit easier because actually in the middle of this space it cannot be like 10 kilograms, right? So how big is our brain by weight? Do we have any answers, guesses? It's not 5 kilograms, by the way, so 2 kilograms. 200 grams? 200 grams is like a pineapple. It's an apple or something. Yeah, I mean, 2 kilograms is actually if a person was having hydrocephalus, maybe they reach 2 kilograms. Yeah. Yeah, so hydrocephalus is a condition where there's water accumulating, but this is quite close. And the answer is around 1.5 kilograms. So this is like how big it is. And there's a bit of variation across different sexes and different age groups. And when you're aging, your brain weight changes. And interestingly, Einstein had a smaller brain by weight, but his neurodensity was greater. So kind of amazing. It's the simplest one, I think. And the second question is very interesting. It gives us insight about how complex the system is. Number of neurons in our brain. So neurons are a type of cells that are nerve cells. And any guesses on this? For those who know the answer, please do not answer it, or just, yeah, Philip, please. Okay, I thought you knew the answer, but you got it wrong. Yeah, but I think you just did it on purpose. Yeah. So the any other guesses before I've been moved? It's quite close, actually, quite close. You said 83 billion.
Like, what would somebody who have 83 billion, what would the medical condition?
Oh, well, there's no medical, well, potentially there's some medical conditions, but that's a really close guess, actually. Do we have any other guesses? That's already close. So let me review the answers. So maybe you cannot count how many zeros there are, because I just want to make an impression of how large this number is. 86 billion neurons. And it's hard to make sense how many neurons that is. It's just like, it's hard to make sense how rich Elon Musk is, right? So we have all these numbers following so if you're a really bored person, and the only thing you do is counting neurons in your brain, you count 1 neuron per second, it's going to take you 30 years to go through the first billion of neurons you have, and take you more than 3,000 years to go through all the neurons you have in your brain. That's how many neurons we have. And in addition to that, you may know that, but maybe you don't know, but the neurons, they talk to each other on a daily basis, a second, millisecond basis. So they talk to each other so they can transmit information. And among these two neurons that are connected, there's 1,000 to 10,000 contact points. That's how complex the system is. So these neurons are constantly activating and talking to each other, although in a very sparse way, but it's already rendering the brain a really complex system, given the number of units, the basic processing units we have. So the next question, I mean, now we have this number of neurons, and we want to put them into small baskets depending on their functions, how they look like, where they are in the brain, and what they're responsible for. So there are more than how many types of brain cells? I mean, any guesses? How many? Go ahead. Yeah. More than, of course, yeah, there's more than one. That's good. Yeah, we have neurons, we have glias, 15. That's a good estimate. Any 300? Kind of getting closer. Yeah. Do we have any better approximations than 300? 450? Okay, we are kind of increasing linearly. Now, do we have any? Sorry, excuse me? 8,000.
I think we'll be at 8,000 at some point. This is as of 2023. We might have more in the future because we are constantly discovering more types of brain cells. And as the day goes, there's potentially one more type of brain cell discovered, potentially named after a person or something. I really want to have a brain cell named after me. But anyways, yeah, so this number will be evolving and doubling and potentially tripling in maybe 3 decades or so. So next question, it's a really intuitive one. So if you recall the law of the United States of America, you cannot drink until you're 21 years old. How many years does it take for the brain to fully mature? Like if you're 80 years, okay. Or until someone dies. Of course, the point we are dying, we are the maturest by, yeah, we know the most, we forget, of course, we forget. So, but what's a more reasonable estimate? Yeah, 100 years, not 100 years, but pretty much 25, yeah, makes sense. So by the time, more than 20 to 30 years, if you were really like doing your PhD without any breaks, by the time you have a PhD, you're pretty much, your brain is mature. Yeah, very, very much. Yes. So, nice. But anyways, these are, I forgot, this illustration is some kind of neurons we have. We look at how these the neurons are different by the shape and by where they are on in the cortex, right? And last one, so for those who have an interest in racing cars or cars in general, you may have a better guess of are these the transmission, how fast the neurons talk to each other, is it faster than cars racing or something? Do you have any guesses for that? But again, it's a really confined space. Like this is how big my brain is. And the neurons are really transmitting information at fast speed. As a hint. So do you have any guesses for this? How many, excuse me? Yes, it's more like a speed. So yeah, like a meter, a nanosecond or something. Excuse me. 5 meters per second, I guess. Kind of fast given how small. Yeah, please. Yeah, but this really depends depending on the insulation condition across different types of neurons and the innate properties. But it's kind of a good estimate to go from the speed of electrical impulses. Yeah. Any additional guesses so far? Yeah. Okay. Yeah, I mean, you can't say people where, but you can. There's a really interesting way of simulating the neurons you have in the brain as wires and stuff. We can talk more about that. But anyways, it's very fast. Turns out to be very fast. This is the speed of a commercial jet. So it's faster than like how the car can travel on the road, I guess, which is quite interesting. So given the number of cells we have, the complexity of functions it can carry and how fast they're talking to each other, this is a machine that never really goes to sleep, even in your sleep. So it's very hard to understand the brain, and some neuroscientists are really going negative nowadays, saying it's never possible to crack the brain. I'm not sure about that, but studying it gave me a lot of joy. Still, I wanted to make a point clear that neuroscience is something that's really new. And by the time I was attending college in the 2010s, there's no major in neuroscience in the United States, or maybe there's one or two schools, but it's for rich kids. So anyways, yeah, so I mean, how young this subject really is. Over just about 100 years ago, we did the first recording of electrical activity in the human brain. So now it's a really common diagnostic and brain monitoring to use the electroencephalogram on the human. So if you break it down, electro means it's just electro, and encephalo is the brain, and the gram is like to graph it. So the first time it's done by a German psychiatrist, Hans Berger. That's really barely 100 years ago. We had the first electrical activity recording from a human. And a couple of decades after that, I don't know if you know the word lobotomy is something very notorious, obnoxious, any human considered by the current standard. But still, 70 years ago, we are still awarding this procedure a Nobel Prize in physiology and medicine. There are a lot of debates regarding if this award should be rescinded. But I mean, to give you more context, lobotomy is a procedure that kind of resects the connection from and to the frontal lobe. If you were performing this on a person, you're pretty much rendering them in a stupor state. Like they don't have their self-conscious anymore, they're behaving like kids. And yeah, it can be, it's written in a lot of novels or sci-fi or dystopia movies described as something that really makes a person lose whatever he or she was. So yeah, up until 70 years ago, we're still giving prizes, a Nobel Prize for this kind of procedure. And so then like how new neuroscience of the field generally is. And a couple years after that, we had the first precision instrument to look at the brain cells. This is the first time really we settled the debate between the reticular theory, which claims the brain is kind of a bowl of soup, everything's all connected, there's no boundaries at all, against a neuron theory by Cahal. So neuron theory claims that there are individual units in the brain and they function individually, despite there are a lot of communications going on. So this is the first time this debate is settled. So nowadays in pop science, everyone knows, okay, there are neurons in the brain, or cells in the brain, there are individual cells, but that's some kind of relatively new knowledge we had acquired by using electron microscope. Yeah, so it's a very young subject. And now, this is the last one I have regarding how new the subject is. Introduction of functional magnetic resonance imaging in humans. We know when we are going to hospital, sometimes we do this MRI procedure to get an image, internal image of how the brain or the organs look like. And functional MRI is a really cool thing by Popney of saying that if we send a person into this machine and we perform tasks, we can know which brain areas are activated when a person is performing a certain kind of task, thanks to MRI. And now, despite its great popularity nowadays in pop science and all this media stuff, it's only introduced around 30 years ago. So that's when we started acquiring MRI scans, functional MRI scans in humans. Of course, but thanks to our intelligence, we have been introducing a wide array of instruments to study the brain from across all the different spatial temporal scales. This is an MRI machine, by the way. You just have the patient here and you can scan their brain or other organs as well, but the brain is the most interesting one. So you can also do this procedure on small animal as well to understand their nervous system. So that's called small animal MRI. Yeah. In addition to that, we have talked about the EEG briefly. So this is how it's done exactly. You have electrodes close to the surface of the skull, and you're recording electrical activity going on across different brain areas using this measure and how that pretty much looks like. Yeah, and this does not really connect to the brain, apparently, but people have been recently introducing this procedure to measure how interoception works. So interoception is defined as the way we perceive signals coming from inside the body. The thing this thing works is we swallow this capsule. There's a kind of a vibrating motor inside that. And by the time it reaches the gut, it starts to vibrate according to the interval or rhythm. And a person is supposed to press that button and report it. Because we have the ground truth when it's vibrating and by what rhythm it's vibrating, you can kind of correlate this person's precision of the perception with the actual signal. So this is a really useful tool and has been used in a couple studies to differentiate between people who have eating disorders, mood disorders, and healthy controls. Because these people who are suffering from these disorders, they have altered interoceptive functions. And in addition to that, it's not always possible to always get this stuff to work. And sometimes we wanted to look at the neuron individually, like how, for example, a small cluster of brain cells are talking to each other, or how this is responding to a certain stimulus. This is the way we do it. This is one example of in vivo patching, which means we can have a tissue or animal situated on this platform, and we insert a probe into the brain, directly accessing a cell, which is mostly a neuron, and we have some stimulus and we see how the neuron is responding. So that's pretty much a direct access into the brain and how individual level, how the cells function. And in many studies in neuroscience, of course, it's inhumane to just grab your colleague to, okay, I'm gonna cover a brain open and run some experiments. It's inhumane, or it's kind of inhumane to put a sensor on and without an authorization, just have them run across spaces and record their trajectory. So for this purpose, we have animal models. And this is one of the animal model that's kind of a protocol that's widely used in in neuroscience. And this is recording their behavior in an open area. And it's been serving as a really valid tool for making distinctions across different behavioral subtypes and stuff like this. And we're gonna talk more about it a bit later. Still, given a lot of different parameters and apparatus we can use to quantify the brain health and disease, we have, of course, looked into how the brain is functionally different and structurally different in different disease conditions. I mean, disease is a really unfortunate thing to those who have it, but for the development of neuroscience, there's a great number of patients that contributed to that because of the disease or the surgery they had. This is one of the first and very prime example, patient HM, American person who had a really severe epilepsy in the form that he seized around once in a couple minutes, very spontaneously. There's no way to predict when it happens. So he went through a really exploratory procedure in his, I think, early adulthood years. And this so-called temporal lobectomy really curved out his temporal lobe and a lot of structure associated with that. And as a result, his hippocampus was removed. And then the surgeons who performed the surgery on patient HM found out he could not form new memories anymore. And this is a really critical step for us to establish a direct mapping from hippocampus and memory formation. So this is one example of how we can achieve a mapping from a local lesion to a certain cognitive function. And this is not the only example. There is one more example about a woman whose amygdala is fully impaired and she had no fear anymore. Again, the brain is very amazing the way it compensates for itself because of complexity and plasticity. So this girl, I think her name is Jody, and she had this really rare neurological disorder when she was 3 years old, Rasmussen's encephalitis. So which we don't know why it happens and what's the pathological origin, but they just have this, like the kids from what 2 to 10 years old just have it. It's really rare, but it happens in some people. So in this case, half of her brain is actually starting to die. The brain is attacking itself. And because of that, she again had started to have really severe seizure episodes when she's 3 years old, like once in 3 seconds or something. Sorry, once in 3 minutes. And the only thing they could do to reduce this seizing activity was to remove half of her brain. So Jody is also referred to as the girl who has only half of her brain. And she was interviewed by BBC, I think, a couple years ago. And if you're looking at, this is not her brain scan, it's someone who had a similar condition. And if you look at this from this side, there's no tissue for half of her brain. But still, Jody had a really happy and fully functioning life. And she just has a bit of a weakness on her left side of the body, but otherwise she's very happy and she's fully functioning. Another amazing example is this Chinese girl who was brought to the hospital by her mom because of a complaint of fainting and dizziness. And she's 24 years old by that time, I think 2014. And she had a family, she had a kid, and they found out there's no cerebellum in her. The cerebellum is something that's really central to your sense of posture and balance, stuff like this. We always hear the saying that people who have a weaker cerebellum, they always get car sick and stuff. So this is supposed to be a fatal condition, and people without this part, a very important part of the brain, they cannot survive. But she made it until she's 24 years old. There's some minor delay in her language and motor functions, but otherwise she's all fine. Yeah. And last example we have, which the doctors had no idea what really happened. So you see, like there's a bit of a black hole on the left part of the brain. Does it happen to a patient called Elise as well as her sister? So they both had this condition, but they didn't know that because they were having, again, a normal life. And it's only until Elise was 25 years old, she went to the hospital at her grad school, and because she was always, she has been diagnosed with epilepsy, but she never seized before. So she's kind of curious what happened. And then they found out there's a big black hole on her left side of the brain, and nothing happened. And now I think Elise and her sister Martha, they're in their 60s, but they're very happy. And it's supposed that this part is supposed to be affecting their language function, but they are very verbal and quite talkative, according to what the interviewer said. So this is how amazing our brain is in the face of disease and injury, and how some of the disease or conditions we have can be informing the brain's compensatory functions, as well as general cognitive mapping and stuff like that. And unfortunately, not all the disease come with a black hole in the brain, and sometimes it's really hard to catch it early. And this is, you may have a relative or you may have someone who has Parkinson's disease.
The dyskinesia kicks off. It triggers something in my brain. It's so uncomfortable, it really is. And even talking about it is uncomfortable. I think it kind of triggers memories of trying to eat and the frustration that you get. So like I'm concentrating now, looking at you down the camera. And I forget straight away. you can think you're going to be able to pick up a knife and fork.
Yeah, so the person suffers from postural instability and the inability to manipulate, do fine motor manipulations, and their daily functioning is suffering a lot from this. So why do we leave to suffer until they have really severe motor deficits? The answer is not we want, is because it's really hard to catch a disease as prevalent as Parkinson's disease earlier in its development. So this is a trajectory of Parkinson's disease development. And then mostly when someone's diagnosed, they're at this point of exhibiting motor symptoms that are suggesting great amount of motor deficits and compromised quality of life, such as the two gentlemen in the video. There are earlier symptoms that preceding the onset of these motor symptoms by 10 to 25 years, I think. But take a look at these earlier symptoms, like sleep behavior disorder, olfactory dysfunction means we cannot smell things. Other people have COVID or having the common cold have this problem as well, but you can't say people cannot smell the butter in my kitchen and I'm going to have Parkinson's in 20 years. It's not making sense, right? Depression, anxiety, this is also a common problem for modern day people, but they're not really specifically linked to Parkinson's. Like for these symptoms, it's not very unspecific. Their appearance does not mean we can diagnose Parkinson's disease or predict it based on these symptoms alone or combined, because you may have a lot of other conditions that cause similar symptoms. So that's a challenge. There's no specific diagnostic possibility based on these early symptoms, and by the time we have the motor symptoms appearing, a person is very, very advanced in Parkinson's stage development. So the thing I try to do during my postdoc study is that we wanted to look into some of these prodromal or early symptoms to see if we can use some means like some AI stuff to catch this a bit earlier. And this is what we tried. So look at this. This is the structure that's highly implicated in olfactory dysfunction, called olfactory bulb. It's really situated here in the middle of the brain, like tiny circles here. Yes, and this is a T2 MRI scan, and it's kind of a review of the detailed structure of the brain matters we have inside the skull. The thing we tried first is that because olfactory dysfunction is leading to change in the size of the olfactory bulb, we wanted to understand if we can use this as the only indicator to tell the difference between people who do not have olfactory dysfunction and then people who have olfactory dysfunction, but because of Parkinson's disease or because of something else. So we were really successful in the first aspect. It's possible to tell the difference between people who have no olfactory dysfunction and people who had olfactory dysfunction using the volume of the olfactory bulb. But unfortunately, we cannot really tell if the person had olfactory dysfunction because of Parkinson's disease or something else. The next thing we tried is that we cross this slice out because these are the areas that are really intensely affected in olfactory dysfunction. I mean, we had all these scans from, I think, 45 patients, a really small number. Again, this is one of the things we face a lot in medical sciences. The number is always low. So we get these sections out, and we process them in a way that's making the numbers bigger. And we also add a lot of pre-processing steps to blur part of it or do some occlusion. And we found out that this patch of area here fed into a CNN can lead to detection accuracy of 85 to 88% of Parkinson's disease. Yeah, so that's quite interesting. And we also looked into what is giving rise to this conclusion or this detection. So we looked into this. This is GradCam is a way to visualize where the neural network's looking at when it's making a decision. So it's this area instead of the small balls here, that's the olfactory bulb. So it's actually looking at the area as extending up to the olfactory sulcus instead of the olfactory bulb. So that gives us a bit of explanation of the things we should be looking at. And we can then revise our experimental protocol to be looking at this area specifically in the next experiment. So that's interesting. And olfactory dysfunction precedes the onset of motor symptoms from 6 to 10 years. So if this was successful, can be validated, we can potentially catch Parkinson's disease a couple years earlier. But what about if we were more ambitious and want to go even earlier? We looked at the REM sleep behavior disorder that happens 12, 20 years before Parkinson's disease onset. So for people who have this disorder, 66% of them convert to Parkinson's disease 20 years after the onset of this sleep behavior disorder. So it's kind of a disorder that troubles people a lot. Just look at this gentleman. So this is him in sleep, and he's acting out of his dreams. And he's kicking. He's kind of removing the wares in some certain ways. Yep. He's murmuring and cursing, actually. I'm happy you can't hear him cursing. Like he's kind of a murmuring and in a different language. But he's definitely cursing. And he's kind of punching people in some certain ways. Yeah. Mm-hmm. Yeah, so, I think he's not done yet. But anyways, he was being investigated in a sleep suite, in a sleep lab. So a lot of wares attached to him, polysomnography. So we use this approach to record people's biosignal during their sleep and try to understand what happens. The thing with REM sleep behavior disorder is that precede the motor symptoms very early and can be very troublesome, assuming you have a partner sleeping next to you and you are attacking me in your sleep, and it can be really inconvenient and can cause injury in a lot of cases. So the thing we try to do is we wanted to understand if we can detect REM sleep behavior disorder, from Parkinson's disease patients or from healthy controls, or we can detect mild cognitive dysfunction within this group of people. So I'm just going to present one of the three cases because it's overcomplicated. The thing we did, again, was to take a look into the brain imaging results we had. So this one, you look at this. This is a parcellation as Nina described, how we kind of parcellate or break down different brain areas based on their functions and anatomical architectures. So we looked into these brain areas individually, and we recorded their morphometric information, like the information about their shape. So how thick these areas individually, how large they are by volume, is their surface area shrinked or expanded overall, and is there any local deformation? So how locally this shape has changed, twisted or distorted. This is especially useful for subcortical part of the brain, like within here, because there's no surface area we can measure, and we kind of rely on this information. And especially this area, like within the middle of the brain, is very heavily affected in Parkinson's disease. So this is one example of how our classification went based on these features representing the shape of the brain areas individually. So there is a study preceding this study was done by my collaborator, Dr. Shady Rahel, and he reached accuracy of 70% using single features of brain morphology in detecting RBD from healthy controls. So when we are combining the brain measures a bit more, we reach a higher accuracy of around 80%. And in addition to this accuracy we achieved, we also looked into what are the brain areas that are affected in different conditions, and what are the areas where there's deformation happening, helped us reach this conclusion. So these are some areas that are affected in the condition of RBD, and we looked into these areas individually, and we had a great agreement with other published articles. But of course, there are some other areas we have detected that are not previously reported. It requires further examination. But you can see, like, in the midbrain area, putamen is highlighted here. And this is supposed to be an area that's heavily affected by Parkinson's disease. So with these tools, it's kind of possible for us to potentially catch the neurodegenerative disorder a bit earlier in advance, and we should be highly aware of this kind of non-typical symptom appearing decades after, so we can render this intervention in a more planned and strategic and tailored way. Okay, so look at all this stuff we have. We can use these to collect the brain data. It's very convenient. It's also very smart of us humans to think about all these different strategies in a way of collecting neural data to understand the brain, but there's always areas we cannot reach. For example, when you're trying to record from a deeper area within the brain, from the slides or from the animal directly, you're really going deeper with the electrode, with the pipette you have. So you go deeper, you're crossing more layers of tissue. There's a greater chance the pipette, is mostly made of stretched glass, really fine glass tubes, and they break or they are clogged because they're really thin. And in some cases, the experiments are very hard to manipulate or to control. And there are external factors like human errors affecting it, or it's inhumane to do at all. So the thing we came up with is we have models. Yeah, so the models are inspired by how the real neurons or real networks or real brain regions work, we try to copy that as much as possible, and we validate it based on using this model to reproduce the real behavior and match it with what we see in the experiments. And once validated, we use these models to just do all the manipulations we like to do on this and see how this system behaves as a way to approximate human behavior or human neural network or human neuron cells. Like this is one example of the morphologically detailed neuron. So we do this in animal slices. We kind of do a reconstruction. It's very detailed. There is a soma and all the dendritic branches. And you can look into, like, when you have this model and these highly, finely tuned parameters, you can inject a current and see how this site, for example, the soma reacted and how the electrical potential changes in the dendritic branch. So it's really convenient. You can kind of record and patch from anywhere. There's no limit of time. There's no animal kind of getting expired on the bench. And you can do this anytime I do this a lot in my bedroom. Yeah. And the second thing you can do, the second class of model is that you put all this stuff together. And you have a model then. You have a model of biologically plausible neurons. And this way you can study their interactions. And you compare this with the neurons that are not so detailed and see if you can add a bit of a plausibility to the behavior you're observing by adding biological details. The third class of neuron, maybe some of the machine learning experts here, we know, is the recurrent neural network. And there's no replication of the neuronal details at a single neuron level, but you can replicate, for example, the ratio between excitatory and inhibitory cells in the neuron. Of course, there are two types of neurons if you're just classifying by excitatory and inhibitory. The ratio, and you see how that changes network behavior or connection probabilities across a single units. This is another type of model, which is not really based on the numbers or probability we're having within the unit, but this is based on the spiking behavior of a neuron. The spiking behavior innate advantage is that there's a timing-related information. For example, when you're stimulating a neuron, there's a cluster of spikes, and it's really highly time-locked. So this way you can represent more biologically plausible activity in time domain. So the next thing we have, this is not all the model we have access to. This is just some representation, but this is a mechanistic model representing how these different types of neurons are connected within the basal ganglia. So this represents the connection probability. It's really modeled after the animal's basal ganglia system. And this model is really convenient the way you wanted to study how health and disease conditions affecting the brain's behavior. For example, if you have a certain condition that, so this is the dopaminergic neuron. A lot of dopaminergic neurons die in conditions such as Parkinson's disease. You can just reduce the number or reduce the connection probability, whatever you think is the most plausible thing to do, and see how that affects the network behavior. So we have this great amount of tools to investigate, in silico, the behavioral system and the single cells. And this is one example of how I did it. So I used to have a video to pair with it because it's really hard to explain, but the video doesn't work. So I just try to explain it by myself. So this is a box. If you just saw the animal setup, there are two boxes. So you just have an animal inside each one, right? It looks like really boring because there's a box and an animal, and what can you do with that? It turns out it can be quite interesting because the animals, they're prone to investigate a lot more, like exploring the environment a lot more actively when they are first introduced to the environment because of novelty. And in the meantime, if you're having like food rewards scattered across, you can use it to investigate their navigation patterns or spatial learning patterns. So you just have this box, and these are the fine lines are the trajectory of their ambulation, like how they walk within this box. And these readouts are the interesting part. So there's a class of neuron called grid cells. So they call grid cells because their activity pattern is really going in a hexagonal, really regular patterns. So you see it's approximately hexagonal, and there's a certain, so this grid cell in the brain of a mouse will be having higher level of activities when it's crossing all these dots. And for different grid cells, it's also going to have this regular hexagonal pattern, but where it's more active, it's going to be a bit different. Yeah, so this is supposed to be one of the internal metric systems we use to mark the space. We have an internal sense of how much we have pretty much been walking and in which direction we are walking. So it's leading to a great and very interesting behavior called path integration. Because the animal, like when it's walking like this, although it's very detoured and very kind of a twisty ways walking, it has an internal sense of how much approximately this animal has ambulated, and then it's gonna go in a straighter way on the return path. This is all thanks to the internal metric systems done completed by the grid cell. And there are some other couple of class of relevant cell I'm going to introduce a bit later. But let's see if we can simulate this behavior and put some kick to it. So this is a neural network I used to simulate it. There's no need to remember all the details. It's just recurrent neural network. It's all simplified on the drawing. But instead of really training based on the coordinates we have, of course, it can have coordinates across this board, right? Because it's a virtual space. Instead of training based on coordinates, we are training based on the activity of those cells. So based on the metric system, reconstruction of the internal metric system we have within the brain, we are training based on the cell activity without explicitly using the coordinates. We had no idea if this is gonna be successful. So this is quite inspired by a design by, I think, a group of people from DeepMind. In 2019, we just further did. This is a vanilla model. We just want to see if we can perform this learning of spatial navigation, based on the cell fairing pattern. And the second model we did is something I really like. So I like this class of things called neuromodulators, and all my colleagues hear me talking about it every day and every day. The neuromodulators are supposed to be something that facilitate the communication, flexibility, and adaptivity of your nervous system. It can really act across different spatial temporal scales. So because it's across scales, we're really adding this learning rules inspired by neuromodulators at single cell level, the fully connected layer level, as well as the whole network level. We want to see if this so-called facilitator of neural communication and adaptiveness can really facilitate learning in this example. So 2 model versions. Yep. So the first observation we see is that after along the training, we had more training steps. We had the lower the error is, and the error is just calculated by the difference between the training trajectory and the actual trajectory we had. So this error slowly went down quite fast at the very beginning, but kind of plateaued in the version where there's no neuromodulation component added. But still, there's effective learning, of course. It learned to a quite efficient level of navigation. But if you're having neuromodulator inspired rules, the learning kind of continued in a non-plateaued way and went on, I think, until 1,500 steps and goes down like this way. So the 2 observations is that the first one, we can really use this implicit training by the cell activity patterns like these to mark the space internally. And the second thing is when we're adding some neuromodulation inspired rules, it's possible to train even faster. So what's going on behind the scene? When I explained the network behavior, so we looked into the individual cells. We not only wanted to have a conclusion, but how we reach this kind of behavior. So this is a really nice representation of how, look at this. The different cell classes, so this is how individual cells in the fully connected layer are having this activity pattern over the whole field. Let's see, at the beginning, it's really kind of a noisy pattern. We can't tell anything, but then over the training of, I think, 200 training steps, it's kind of emerging to something super specific. Like this, even like this. So this is a biological cell people have patched from the brains of, I think, rodents. And these are the simulated ones we had after training. Look at this, it's slowly converging from some kind of noisiness to these very specific defined patterns. And we wanted to understand if adding these neuromodulation inspired rules is really doing anything to the behavior, but as well as the single cell level activity. So we just took this grid cell, as we just talked about. We studied that a bit. So again, this is a really simplified description of what we did. We also did something additional to that. We looked into how active this grid cell is across different fields when we have this vanilla model and when we have this different neuromodulators injected. So the thing is, when you have no neuromodulation, just go like this, and the variability is really small. And we have a neuromodulation. I mean, of course, the activity goes a lot higher, but the thing is there's a greater variability. So there's greater variability in how active the cell is across different spatial domain. And there's no confirmation why this is more helpful, but the computational hypothesis by a lot of people suggest, well, not a lot of people, maybe like by 4 or 5 different groups of people in there, that's a lot of people sometimes, suggesting that when you have a greater variability, you can really make more numeric combinations to represent greater amount of patterns. So I guess, hopefully that's straightforward. As a potential benefit of having this variant field variability. Yeah, so that's one example of how we can use artificial neural networks to approximate the brain's behavior in some certain way and explore it down to the single cell level, match it to the behavioral level changes. And let's look back at the many ways we have investigating the brain, like this. So electrical activity from electrodes recording is really go at the whole brain, the systems level, right? And also we have a really fine, we have a fine temporal resolution of milliseconds, the EEG activity, and this is also done by recording of EMG and EEG, so electrical signals in the muscles as well as in the brain. So they're at a really systems level with a high temporal resolution. And for the MRI we have, which is like this, we have, again, like a more system level resolution. Of course, you can, with the greater resolution you have on the MRI scanner, you can zoom in a bit more. But of course, the temporal resolution is not so ideal until recently, it's still not so ideal. And then we have, for example, patching from single neurons. And then you're recording from a single neuron behavior instead of the whole system. There are so many levels in between them. And the time precision can be really adjusted in this case. So since 1988, if we're comparing these two graphs, you can also see how much the tools we have evolved over this couple of decades. But again, very despairingly, the investigation into the brain are across multiple scales, and it's really hard to bring the data together across scales to explain why behavior at one level, like such as single neuron level, is contributing to the cognition at a whole brain's level. So the next thing I'm trying to do, or something I have already started, is that we want to have a systematic approach to data analysis, storage, retrieval, model building, and task shaping. So there's a couple of representation of data we are really ambitious. Yeah, there's a lot of type of data. So this is the brain connectivity data done by diffusion imaging. This is the recording from, I think, the rodent cortical column and how these areas are connected. And these are single neuron level recordings of neural behavior based on the neuronal type. So these are different types of neurons. You're going to have all this kind of data in one data bank. Of course, not like across types. But so systematically storing it, we are also doing a bit of an indexing system to look into what are the cell types in each brain region, what are receptors they're associated with, and what are experimental setup, and what are the species we're using. Because there's recently a paper that's suggesting experimental preparation for the same group of animals can really introduce a systematic deviation. And there's also like species-specific effects of experimental manipulations and property of the same group of cells as well. So the thing we'll do is that we derive insights from these data and the higher level insight mathematical. Also, we are gonna do some model fitting as well, and modeling of this neuron's behavior. And we have the more biological constraints to act out on this the models we have. Then we'll also use experimental preparation described in this data bank to reshape and further refine the different behavioral tasks we have, in addition to the machine learning landmarks, so we can, benchmarks, we can use them to test the models we have. So this is a really big step. I'm gonna be working on this for the next half a decade or 5 decades, I don't know. But I really like this approach where we are building biologically inspired neurons, and neuronal models will not be like looking at too much detail. It's gonna help us, understanding the brain. But anyways, here are my 2 directions I presented. The first one is really to understand the brain through AI and multimodality data. Second one is about how we can use these data to do, more neural-inspired models making sense. These are my brilliant collaborators across the world. I'm happy these flags are showing up. And my research has been generously funded by these places around the world. And if you're interested in my research, please come and talk to me. And if you're interested in collaborations or anything, just that's pretty much it. I think that closes the talk for today. And thanks a lot, Nina, for giving me the start. And that's my talk. Thank you a lot for your attention.
Thank you, G, for this very, very inspiring talk. There's already the first question.
Yeah, thank you very much. Your plan is quite big. So you want to build this bank, and you said, oh yeah, it will take you a couple of decades. But so far, they have been a lot of, there's the UK Biobank, there's the Human Brain Project, there's a lot of different data banks of diseases where you can also like add tissue and have a certain behavioral results on other clinical input. Will you work on can you use those for your models to start already?
Yeah, we have some models embarrassed by that, but these are a lot of investigations are humans, or there's a higher level. So there's a class of models called computational psychiatry models, especially about active inference models and stuff like this, highly mechanistic and abstract using this kind of data. But for mine, I think it's going to be a lot of based on animal recording, because the things I'm interested in, like individual neuromodulators, cannot be so well recorded in humans because it takes yeah, I have to restack people's brain to do that. I guess it's not possible for me. Yeah, but the thing we wanted to bring to this is that we want to create a sharing platform for different labs to come together to share their data on the same thing. Yeah, exactly. So it's really a bit of a neuromodulator driven, because there are different labs researching the same neuromodulator. And there's a study head by Nelson Toda, a really talented researcher in Helsinki, suggesting that all these factors are affecting the results we see. So there used to be two firing patterns or activity patterns in the locus ceruleus neuron. And now when they put all the data together, there are 8 types instead of 2. So next time, maybe there are 32 types instead of 8. But I think this is really important when you have this kind of data. Instead of manually analyzing it, I'm not offending my experimentalist colleagues, because they're really like, they do this manually. I think the way to gain higher level insight is really to have something you can plug and play and indirectly displaying based on all these indices, instead of manually tagging things. So we can create an automated pipeline for going through all the parameters, see how that affects the results we have. I think that's really important to gain the next step, the high level insights. Hopefully that's sufficient. Thank you.
There's even one more question, and then we go to the panel.
I'm a vegetable scientist. 2 systems have asked 2 philosophical questions. The first one is, when you model something, you have always to take in, at least in my philosophy, we should take care of Karl Popper's falsification. Now, there will be for sure models which do not predict the outcome of the animal, the mouse, the ant, or the human. Then formally one should discard the model fully. What is your point on this? And my second question is also philosophical. Where is the free will, which we believe we have in your models?
Could you please repeat the second question again?
We humans believe that we have free will.
Wow, so philosophical.
Okay, yeah. That we have, but if just where is it?
Oh, wow, that's a really higher level question. So first, the thing of falsification and RNN, yeah, RNN is the center to everything. But anyway, yeah, it's hard to pinpoint. I mean, I'm gonna go from the second question actually. So there is a hot topic in neuroscience, and there's an article going on for 10 years why the most brilliant minds in the world cannot answer question consciousness and stuff like this. So there is a neuroscience experiment done around 2014 suggesting that before we are acting out something, our brain didn't know it. So they use this as evidence against there's free will. So we thought we were deciding everything. We thought we were making careful decision making, but actually our brain has decided for us. It really depending on how you're investigating it. I mean, I think there's going to be more experimental evidence, for example, at a higher resolution. It's not only the time, the time in the time course, if something is decided first, is not free will anymore, because it really depends on if you think the brain as an agency for free will or not. I think it really depends on how you define it. So the first question, the falsification and the complication of modeling. Yeah, so there's a popular saying, I think already brought up by Chris in the last week's lecture. Well, the last edition of the lecture, all models are wrong, some are useful. I actually initially had this quote on the slide, but I thought I don't want to go deep into this. So yes, all the models are wrong. And I'm pretty much saying all the models we have are limited, especially because we don't have the device to run through all the details biologically to do it. So yeah, but there's a difference between intended falsification and the mistake we have because of limitations of the model. So we really have to be careful in the way that when we are reporting the data, the methods we did to do it, there's no falsification, and we are kind of reporting the limitations of the model and what we see that's contradicting experimental evidence carefully, so people know what are the limitations of it. But I just don't dodge the model because it's not reproducing whatever I think. I think now I'm really a big supporter of null result reporting, so people know what we have done, and people will see if they have the same null result if we are reproducing the same methods like we did. Yeah.
Okay, one last question. And
A specific question on the Parkinson research you did. So, have you also thought, or are there indications that there are genetic causes for it? So, can you maybe directly look into the genome of the patients you looked at, and have you looked whether there is something that gives some information on the cause?
Yeah, genome data is not really my domain, but I'm pretty sure there's a 2022 study done by the NIH. They looked into the genome of Parkinson's disease patients systematically, and they found they can be used in combination of other biomarkers as a way to do the prognosis and prediction. And I mean, although I don't really do genetic data, I strongly believe that there are genetic linkage between different forms of neurological disorders, neurodevelopmental disorders, neurodegenerative disorders, because I think a study published in 2021, they talk about the arterial, like the blood vessel, there's a level of similarity across different neurodegenerative disorders. I think it's a really useful tool, and people have achieved high accuracy in detecting Parkinson's disease patients using this kind of tools. So it's already being done, yeah, and still ongoing.
okay, so now I have to drag you out of the neuron level into the ITU level, because we want to talk a little bit about interdisciplinarity. So Nina, please could you join us again? So, oh, thank you very much. So since ITU is the university where interdisciplinary researchers meet and mingle and create more interdisciplinarity, my first question always is, how did you get started with interdisciplinary research, Nina?
It might not be the normal case, but I originally wanted to study something else than computer science and medicine and psychology. So I was like, but I didn't want to become a doctor. I just wanted to, be a researcher. Then I understood if I work with data, I can research in medicine, but I don't need to be a doctor. So I was like, pretty clear, I was like wanting this direction.
So around the corner, so to say. Okay.
Yeah, I also wanted to be a medical doctor of practicing medicine while I was like 6 years old, because that's something I thought led to the brain. But not really, I mean, I don't want to be in a neurology ward dealing with, the conditions every day. I want to be researching it. So I thought it's the wrong direction. And it's kind of easy for me to start this multidisciplinary practice because I changed the major 4 times in college over 2 years, because I really had no idea what was this discipline and where we can study the brain.
Which majors were there?
Initially, mechanical engineering, electrical engineering, computer engineering, computer science. Yeah, because I did robotics in high school, and everyone in robotics went to mechanical engineering, and I really had no idea because I was the first STEM major in my family. Yeah, and
So you went from the tangible to the intangible, so to say, yes?
exactly. Then I was very lucky that I found a cognitive science program while in my master's, and where we had 6 different concentrations or specializations: philosophy, linguistics, cognitive neuroscience, computational neuroscience, and some other stuff. Yeah, that's the first time I came to meet a lot of people from different disciplines, like ranging from economics to arts and design and mathematics. And we really had a chance to talk directly and think about how we approach the same thing differently. So that's how I exactly started. Yeah.
So Nina, what do you think interdisciplinary research is difficult? It seems like it came very natural for you. Do you think there are some roadblocks or?
Well, I guess everything has its challenges. For me, going from as a computer scientist towards medicine, towards explainability, one of the big roadblocks was always, so where do I get the data for the high stakes decision? How do I get the collaborators? How open is the connection between them? It's very different coming from computer science, talking to medical doctors. They're always busy, a very different style of working, very different style of publishing. And to get to know all this is, it makes it when you're just a computer science, at least you know what you're doing and with whom. And with your more broader, in this case in medicine, it is, you broaden not just your own field, you actually broaden how you work.
What about you? I agree with that. I trained as a pure computational neuroscientist at a master's program. Then I went to a medical institution for a PhD, but now doing a MD. I was the one analyzing the data. I was also the one analyzing how machine learning model could help analyzing the data. And the people working with me, medical doctors, they were analyzing the Excel sheets manually. So there's a big gap in the way we were talking, and I wrote a script to automatize it. They were not willing to use it because they thought it could render some errors. So this is a challenging part because you have to convert people across disciplines to gather insights. And in addition to the different ways of working, I think another layer of complexity is that there's a lot of things to learn. For example, I just picked up network neuroscience, and it's a lot of new things to learn for me, and also like mathematical modeling of the neural system. I think it's really sometimes to think about, there's a lot you have to learn. It's kind of making me nervous, but I guess the benefit of doing a PhD is I feel you can learn pretty much things that are there, and on the other hand, it's quite exciting. Yeah.
Well, you both are working on the brain, so that's definitely a very, very exciting topic. And with this, I think you here for being here. I thank our online audience for listening to us. And with this, I would like to invite you for a little drinks and a nice evening. Thank you.